Welcome everyone to the second session of CAPS Talk series. So last time we talked about protein sequence search, and today we are going to dive deep into the search of protein domains. So a general rule, so please keep yourself mute and switch your videos off. And if you have any question, please put it in the question and answer session box. And if you want to discuss something, please put it in the chat box. All the questions will be taken at the end of the session. Over to you, ma'am.
Thank you, Shaila. Hello, everyone. Good afternoon. And we are meeting after a week. And last week, what we had discussed was on various topics such as structural motifs that, from our lab, the experience that we have been having for the past few years. And then it also talked about the interacting motifs, that is, those motifs which are conserved as seen during evolutionary scale, and also interacting spatially so much that they may be important for the structural integrity of the proteins. And then we also talked about cascaded sequence searches, in which we hop from one family to reach another family using pure sequence information and only computer algorithms, wherein we provide each of these hits to become queries so that we can hop in the sequence space. And these sort of approaches are helpful because these structural motifs can remain as constraints and then help the sequence searches to be more sensitive, yet be specific. And also these sort of cascaded sequence searches could be useful because they can then get into the one-time computer intensive sequence search, which we called as GenDis. They can kind of get tailored in. So this looks like a very happy tailor. However, in today's discussion, what we are going to talk about are domains. Domains are often found in proteins, not that every protein should have multiple domains, but let's imagine a protein domain whose sequence information is shown on top, and then they have a schematic of the primary sequence, the primary structure, the lead amino acid sequence of the protein. And lo and behold, if you look at the structure of the same, you can start to appreciate that in this case, what happens to be a homeodomain, and the structure is color-coded such that the starting from the beginning of the sequence, what we call the N-terminal, until the end of the sequence, which is called the C-terminal, this goes from blue through various colors to red, like a Wig representation. And you can see already when you actually we have tried to put it through videos so that you can appreciate the real-time rotation, and you can see that there are actually two compact regions of the protein structure, and these are referred as two domains. And up on the schematic, therefore, there should be one domain shown in blue color, another shown in red color. In between, they're connected by this kind of wavy-looking thing, which is pretty disordered. This is to appreciate that you can have multiple domains in proteins, and they might carry out their own functions, and they may be so structurally independent that they can hold almost on their own. So this is good to know, but let's look at another example, because sometimes these multiple protein domains are required as structural elements in the biological cell, and in the yeast structural protein, this is what happens. And you have actually two domains. The one domain is shown in the front, and then the second domain, which is shown like a pillar. So almost like these are girders in a bridge, and these elements are vitally required for the stability of the protein. In fact, you see not one but three such proteins coming together, and it's called as a trimer. And this is arranged as two domains and three such proteins coming together. Well, what happens in the third example? We'll give you a third example here, which is a hypothetical protein whose structure has been solved by means of genomics initiative, and it's from Plasmodium. And this is looks pretty complicated, and it is, because starting from the N-terminus, a part of the domain gets folded, and then it seems to change its mind, go to the other domain, come back, go back, and this happens many times. And in fact, arguably, that some people may not even agree that there are two domains, but yes, there are two domains. And in fact, these two domains are kind of intricately talking to each other as though they are involved in a salsa dance. But all beauty aside, how does it affect sequence searches? That's the part that we had discussed last week. And back in sequence searches, these matter. This matter because let us take this fourth examples which I have put forward for you. Another two-domain protein. This is very common. This is called as a discontinuous domains. Again, looking at the color code, you can see that a little bit most of this domain in blue has happened, but then it goes to the other domain and comes back at the very end. The C terminus, as it is called, the red color one, together forms that domain which is in blue primarily, suggesting that this is a discontinuous domain. So you can imagine how much of issue can arise when we have pure sequence information on top and we are relying on computer algorithms, whether they are guided by interacting motifs, structural motifs, or cascaded sequence searches. Each of these little bits of the sequence have to be convincingly connected to these structural fold, and there are two domains here. So this is going to be a huge sort of a challenge when it comes to sequence searches. However, this is not the only type of challenge. And you can see now this Taylor is no longer happy because not only he is faced with discontinuous regions of the domains with respect to sequence, but if you actually look at the huge expanse of sequence database, remember the very first slide that we had put up, the number of protein sequences are much, much more than the number of protein structures. That's the one. The UniProt is a kind of a very reliable database, one of the very reliable database where you have 100 million sequences which are contained. And imagine if people have tried to connect with other databases that hold all the domains, well-known domains. And one such repository is called PFAM. And we will come to that later as well. The coverage, starting from these 100 million sequences, how many of them do connect with the PFAM database of domains? One gets only a 3/4 coverage. And in fact, even those that get covered, you don't get to see domains throughout the primary structure or the amino acid sequence of the protein. And you can see maybe one domain, and a large majority, nearly 50% of the sequence remain what are called as unassigned. And this might happen either because we are going through strict thresholds, and these are after all computer algorithms. So you have thresholds everywhere, and maybe these connections are just missed because they are borderline cases. And also this might happen because the evolutionary divergence is such that the lactamase domain in this example is very well conserved. But the C-terminal tail region, which actually harbors a domain, may be independently evolving at a very faster evolutionary rate. And therefore, you're able to see this connection using computer algorithm, but you're unable to see this connection. So this is the sort of scenario that happens when you are starting to appreciate domains when we perform sequence searches. So what I'm going to do in the next few minutes in this talk is to present to you 4 stories. First story is our approach of trying to still bring back the borderline cases. And this is geared by a server, a protocol which we call as Pure. And then following that, we are going to look at another algorithm called ADAS, which will provide a coarse-grained approach to appreciating proteins as a string of domain. And third story is going to be on our long-term journey on one of the signaling domains called the tyrosine phosphatases. And finally, the fourth story will be presented on a similar sort of genome coverage, but on a very interesting other signaling domain, which is the toll-like receptor, and one of the intracellular domains in this. So these are the 4 stories, all right? And coming back to this borderline cases, what happens is we are trying to actually take one sequence, the amino acid sequence of the protein, and then using computer algorithms and trying to search for possible domains in this domain database, which is a PFAM. And the PFAM 24th version had 12,000 such mathematical models, hidden Markov models, which contain all the permitted amino acid variations that happen for those families. However, the more recent PFAM 35 version has close to 20,000 such mathematical models. And soon, or as we speak, this PFAM is going to be taken over almost by Interpro, and they work together very well. And Interpro is kind of an integrated domain database. However, the question is the query sequence is about 650 amino acids long, and then we are able to assign only half of the protein sequence to a domain. let us say, a transketolase. So how can we improve this, and how can we begin to attribute or make more connections between the sequence and structure? And for this, what we have proposed in the pure approach is to use the distant relatives, namely the homologs or the relatives that you find by simple sequence searches for the entire sequence by simple sequence searches like a SciBlast, and where you do not worry about the domains for a while. And once you find relatives such as for protein X, there is a relative called protein Y, then we concentrate on this unassigned region for which you could not attribute a domain and see whether there is reliable similarity in the amino acid sequence to that part of protein Y. And once that happens, you then forget about protein X for a little while, take protein Y and go and search in your PFAM database, and you'll be surprised to know that the protein Y can behave slightly differently so that it harmlessly is able to find a connection to a domain C. And that means now we can trace back and say the for protein X in the unassigned region is likely to contain domain C. So it's what can be viewed as a function annotation transfer, but this happens at the domain level. And this has all been geared by one of my students, Chandrasekhar Reddy, who had joined our lab around 2006. And he also happens to be one of the first dual PhD students. That is, he earned 2 PhD degrees, one from here, from India, another from La Jolla, that is Reunion Islands, and therefore he also has a co-guide there, Dr. Bernard Hoffman. And as we speak, we can appreciate that Chandrasekhar is in fact with us. And he's going to actually tell you the rest of this story as a person who has, as a student who had actually worked in this project. So Chandrasekhar, please.
Good morning, madam. Okay, excellent. Thank you so much, madam. It's great seeing you after a very long time. And thank you for giving me this opportunity. I hope to see everyone after the talk. My name is Chandra Chilamakuri. As madam said, I was doing PhD in NCBS between 2004 and 2009. This work is one of my partial work towards my PhD. As madam described the overall problem, when I started my PhD, there are considerable number of sequences in the databases, but a significant number of them were not functionally annotated. So our task that time was, can we improve the annotation of those proteins? So that's the big idea. So this is already madam has showed the general idea behind the approach. So what I did was, I had taken that approach. I applied on one protein family, adenylyl cyclase protein family, and eventually I tried on the genome level by taking Mycoplasma gallisepticum as an example. Okay, so first at the protein family level, so I started with adenylyl class 3 adenylyl cyclase family. So essentially there are a number of proteins, but some of them are well annotated, some of them are not. For example, here there is a domain A and B, but there is a long unassigned region between them. With existing methods that time, there were no annotations can retrieve from databases. So our task was using indirect search methods, can we improve that annotation? So I started with 329. Then after filtering, I left with around 250 unassigned regions. Then by looking for homologs in the non-readable database, then using HMM, I assign the domains to remote homologs. Then those domain assignments bring back and assign in those unassigned regions. So one functional implication is that, so to begin with, this was the protein we had. So this is a protein ID I have given, and there were two domains, HAMP and adenyl allyl cyclase, because my working family is adenyl allyl cyclase. Every protein what I'm working should have adenyl allyl cyclase domain. But problem is, this is a kind of incomplete protein. We know that this is a signaling domain, cleaves the two phosphates from the ATP and starts the cascade of signals downstream. And HAMP is a kind of present in the membrane and the N-terminal unassigned region. There must be some domain that must be capturing some kind of signal from outside the world and relaying that information downstream. So we know that this is incomplete. That's why this is unassigned, reasonably long protein, long region. So using our approach, we were able to assign a new domain called cache. So now this assignment is completely makes sense, because cache is a kind of signal sensor. It can bind to calcium, other small molecules. It's a kind of chemotaxis sensor. So then it translates that information downstream to this adenyl allyl cyclase domain. Then it creates some kind of downstream cascade of signals. So that is one brief example, the implication of our work at that time. And we also applied this approach on the whole genome level by taking Mycoplasma gallisepticum. So when we started this work, if you look at the Mycoplasma gallisepticum proteome, so only 15% of 15% of proteins or domains are closely related to other known domains. Some of them are missing, some of them are very, very distantly related. For example, this is the structural point of view. But if you look at the domain, PFAM domain point of view, so this genome has around 500 protein sequences. And with that, so when we search in the database, around 400 of them have at least one hit. That means that that protein may have at least one PFAM domain. But interestingly, it's only 56% of protein residues are covered with some domain. Remaining 44%, we don't know any functional annotation of those residues. So, so we did exactly same method. Take all the proteins from Mycoplasma gallisepticum, look for remote homologs, then assign domains to those proteins. Then, if there are any domains we found, we can map back onto the original sequence. So, although we have 620 unassigned sequences to start with, so after filtering, for example, length, coil to coil, and low complexity regions, etc., we left with 359. I take that one, look for homologs, then we assign domains. So we were able to assign 62 domains onto Mycoplasma gallisepticum genome. But we still left with 63, even through remote homologs, we were not able to identify because they have passed all those conditions. Probably this may indicate new domain families. That makes sense because every species is unique, although they take the species originate from the previous species. They take some of the pre-existing domains, and they modify some of the domains for their needs. For example, Mycoplasma gallisepticum is this is a pathogen on the chicken. Some may be on humans, some may be other species. So I have shown one example here. It's around 600 base pairs long. By using our approach, we were able to identify kind of RNase-like function with the two domains, domain 1 and domain 2. But I was curious, that time there were no assignments. We assigned this. Now, yesterday when I was looking at the interprobe, for example, here. So this is the, now there is a huge amount data generated after 10-15 years of my PhD. Now, if you look at interprobe, they have come up with a similar annotation. For example, RNase, ribonuclease activity. We were able to predict this activity almost 15 years back using our approach. So that indicates that we have used correct approach for the annotation. With that, to summarize my PhD work, so we have taken indirect search approach to annotate the proteome. We were able to identify 67 new domains in the 329 class 3 adenylyl cyclase protein family. We were also able to apply same approach on the Mycoplasma gallisepticum genome, and we have assigned 62 new domain relationships. And we also predict that there may be new domain families, which they did not have hits even with the indirect approaches. With that, I'm going to hand over back to Madam.
Great, that was a fantastic story, and it looks and sounds much better from hearing it from Chandrasekhar. Thank you so much, Chandrasekhar. And to the audience, Chandrasekhar is now a senior scientist in Cancer Research Center in Cambridge, and he's been. We are happy to have you online, and please do stay on because the questions at the end.
Yeah, sure. Thank you. Thanks so much.
I will take over now.
Yeah.
And then we will continue. In fact, what I wanted to say is even a 10% improvement in function annotation at the whole genome level, even in bacterial genomes, is considered to be very significant, right? Chandrasekhar was able to bring in function annotation for 60 out of 600 and odd genes in that bacterial genome. That is actually a very nice thing to happen. But let me just go on to tell you, for instance, having gathered additional domains is nice. But like I mentioned in the previous talk, our interests don't usually stop with just looking at one genome, right? We had been looking at multiple genomes, 100-odd genomes using the structural motif constrained approach. And then I also explained our kind of a GenDis, which caters to a large number of domains. So when that happens, you can imagine that you are left with a huge amount of domains to comprehend. And this is not anymore something which you just sit down on the table and look at them manually. Of course, there's a lot of joy still selecting a pair of genomes and one family like adenylyl cyclase, sitting down with the domain architectures and comparing them. But in reality, our bioinformatics analysis are such that statistically they become significant if you can manage to look at a large number of protein families, preferably of unrelated folds, and into a huge number of genomes, so that we can draw some inferences that stay put across these variations. And with that sort of philosophy in mind, what we next decided to do is to come up with an algorithm which we called as ADAST, Alignment-Free Domain Architecture-Based Similarity Search. And this is pretty intuitive. What we basically do is to consider proteins as a string of domains. And then already I was telling you that one domain, like a beta-lactamase, may be highly conserved during evolution, whereas the other domain, the coexisting domain as we call, may be rapidly evolving so much that it is a distant relative now, so you are not able to see it. So these sort of preferential and different rates of evolution of individual domains used to bother us. And we used to wonder why let this sort of differential evolution come in the way of our comparing proteins that are similar across genomes, equivalent across genomes. And for this, we decided to consider proteins as a string of domains wherein we temporarily switch off the similarity across domains, across the proteins. And so long as there is a domain present, we put a kind of a unit there. And then it helps us to compare proteins and find out, for instance, having known that there are domains, how similar are the order and the presence of various domains that are contained within two genes that we think are fairly similar. And this is referred as what is called as a domain architecture. And Divya Shyamla Devi, who had joined as a PhD student in around 2005 in our lab, started to then look at them in a triplet sort of an approach where she will virtually, her algorithm will sit on a central domain and then look at their neighbors and see how similar they are across two proteins that we believe have at least one domain in common. And these were all then scored up. And next, I would like to invite Dr. Advait Joshi, who worked with Divya. And Advait was then a PhD student who joined the lab after Divya. So may I request Advait to come online and explain this part of the story, namely the scoring and how it kind of gets transformed in the form of a tree diagram that we can appreciate. Advait, please.
Thank you, Mark. Thank you for introducing about the ADAS. So, may I request next slide, please?
Yes.
Yeah, thank you. So, ADAS is an algorithm where we look at proteins in form of their domains, and these domains are organized as strings of these domains, which is termed as domain architecture. So here we do not look at the protein sequences. Excuse me. Instead, we look at these domains as in a coarse-grained manner, as these beads on this string kind of a way, and then we see how these domains can be, or the domain architectures of two proteins can be compared. So ADAS basically what it does is when we give a bunch of domain architectures as input, it considers a pairwise assessment among all these domain architectures, and then generates a distance matrix based on the domain architecture distance score for the provided input domain architectures. So in this, we are completely zooming out of the protein sequence and looking at these domains in an objective manner, and how different events which are found in these protein domain architectures are scored. Next slide, please.
Yeah.
So, yeah. So what we get is there are several events. So we break down each domain architecture in form of triplets of these domains. So consider there are several domain architectures mentioned on the right hand side of the screen, ABC, ABCD, and so on and so forth. So what we can do is break each domain architecture in form of triplets, and while doing this pairwise comparison, we check for these triplets wherein we see the central domain. Now, if the central domain is matching, it will have a complete match score of 1. If then you can see the next event, the green color circle, where the central domain is matching, but within that triplet there is also a duplication event. So nature has selected to have that domain in form of a duplication. So as a repeat over there. So that is scored as duplication score of 2. Then you can see the orange circle, which says that the central domain is matching, but the neighboring domains have shuffled. They have changed their places. So there was a shuffle happening in the domain architecture, in the sequence of these domains, how they are organized. So that gets a shuffling score of 4. And then the yellow color circle, you can see that the central domain is matching in those triplet pairs, but the neighbors are not matching. So it is an incomplete match. So we score this as an incomplete match score of 6. And finally, the gray circle at end, at below, you can see the central domain itself is not matching. So this receives a score of 12. Now what we do is check for all such triplets while comparing any of the pairwise domain architecture comparisons, and we add all these scores together, normalize it based on the lengths of this domain architecture, and then we arrive at something what we call as the domain architecture distance score. So this domain architecture distance score is a pairwise distance score between two domains and two domain architectures, and this is used for further clustering of these domain architectures. Now one must be wondering why, what is the significance of these domain architectures? This domain architecture distance score when we get these different events, as in complete match, duplication, shuffling. So we actually went to entire, we took all the domain architectures present in entire PFAM, and we divided them in form of triplets. So basically there were 13,000 with one domain which we excluded, 37,000 with two domains which also we excluded, and only those domain architectures which have more than 3 domains, which were around 90,000. We selected these domain architectures among the total number of domain architectures present in PFAM, and which gave us like more than 400,000 triplets, among which there are 56,000 unique triplets. Now, if we see these unique triplets, we can see that there are lesser shuffling events than the duplication event. So this gave us a hint that duplication should be scored lower than the shuffling event. That's why in the scoring scheme, we have a gradation where duplication is scored lesser than the shuffling, and then the no match receives the highest score. Since this being a distance score, higher the score represents, the higher the distance between, or less similar these two domain architectures are. Next slide, please. So you can note over here. So for a given triplet, there can be a complete match, which is on the right hand side, and that is scored as 1, which is the lowest. Whereas it can be a no match, which is on the left hand side, which has the highest score of 12. And then this empirical scoring scheme, what we followed, actually follows the trend presented in PFAM. Then we assess all the domain architectures present and all these triplet events. So a partial match gets a score of 6, shuffling as a score of 4, and duplication as a score of 2. So we followed this grading in this scoring of ADAS, and we implemented this on several data sets. One of them being the protein tyrosine phosphatases, which our lab has already been looking after in multiple projects. So if you see the protein tyrosine phosphatases, basically these are phosphatases who phosphorylate the tyrosine residue. And when we checked into the PFAM, there were 514 unique domain architectures. You can see on in the image on the right hand side that the red color circle is the PTP, which is the protein tyrosine phosphatase domain, and it combines with several domains which are marked in different colors and shapes. Now some of them have the purple colored one TM mentioned, that is the transmembrane region. So such protein tyrosine phosphatases are receptor type tyrosine phosphatases because they have a transmembrane component over there. Next slide, please. So over here, these phosphatases can be broadly classified as cytosolic phosphatases, receptor type phosphatases, and dual specific phosphatases. Again, dual specifics are some special cases where they phosphorylate not only tyrosine but also serine threonine residues. while I mean, similar to the another class, which is serine, threonine, phosphatases, they have their own class, but this dual specific to both the things. Now, when we look at PFAM and these 514 unique domain architectures, the phosphatases are represented by several domain notations, like you can see in the table mentioned over here, y_phosphatase, y_phosphatase2, and so on and so forth. And they have their own number of sequences which represent this domain, as well as the domain architectures in which they take part. Now, what we additionally try to look into is assign each of these domain architectures, these 514 unique domain architectures, to the kingdom they belong. Kingdom per se, the taxonomic kingdom, whether they are eukaryotic protein, they are bacterial, archaeal, or viruses. So this was a very coarse-grained way to look at the taxonomy, but one can go even further and dive deep into the taxonomic levels for assessing these domain architectures. So we use this domain architectures, 514 unique domain architectures, which actually correspond to 26,000 sequences. So if you imagine aligning these 26,000 sequences, then proceeding for phylogenetic tree reconstruction, it is a very challenging job. First of all, when you have so many sequences which have so many different domains combining with each other. So next slide, please. So in ADAS, this is the beauty of this program, that it helps us to look at such a huge data set in an objective manner. So you can see the cladogram on the left hand side, where the receptor type phosphatases have clustered separately. The cytosolic mentioned in the magenta color are clustered separately. Low molecular weight phosphatases, and so on and so forth. There are so many different types of phosphatases which got clustered separately. Additionally, we also found that they could even subcluster according to the kingdom they belong to. For example, the gray marked clades are those which are coming from bacterial kingdom, whereas some unusual ones came which are like viral and eukaryotic dual specific phosphatases. They clustered together, as you can see in the cladogram on the top right corner. So these are the cues which we obtained from the entire data set, and ADAS could handle it because it was in a coarse-grained manner. Now, this was for entire PFAM, all the phosphatases found under the sun. But one can even go to a specific organism. So as you can see in the next slide, where we consider all the human protein tyrosine phosphatases, and you get even further resolved cladogram for these phosphatases. So there are 56 domain architectures present in human protein tyrosine phosphatase dataset, and they are divided as the receptor type, cytosolic type, dual specific, and the other two types, that is low molecular weight and PTPA. And they all separate. I mean, they all they all cluster very nicely in a separate place, as you can see with the different colors coding. So this is the highlight of the ADAS algorithm, which can handle these domain architectures, huge data sets, as well as the focus data set if you want to go across taxonomic level, as well as throughout entire database. So what we consider here is a domain-centric clustering, which allows us to objectively look for a particular domain and its and the domain architecture it takes part in. So that's how we have used the ADAS algorithm for classifying or clustering the protein tyrosine phosphatases, and there are so many other domains which can be used, domain architectures in which it can be used. So thank you, ma'am. You can take up from here.
That's great. That's great, Albert. Thank you. And in fact, we are just using the dendrogram to our advantage, because we have inscribed what usually goes in as a sequence dissimilarity score, and instead of that, we have placed in the ADAS score. And the phylogeny is purely coming from the domain architecture, not worrying about the extent of sequence variation. So only the presence or absence and the order of the domains matter. So this is sort of a coarse-grained approach, is pretty powerful, as explained very nicely by Advait. So that was our second story. The first story was on the pure, how to assign regions to unassigned regions, domains to unassigned regions. And the second was this sort of an ADAS approach by which we can do coarse-grained analysis of domain architectures. And the third story, which is pretty continuous actually, is to understand our journey on the analysis, the sequence analysis of the very important, one of the important signaling domains, namely the tyrosine phosphatases. And this is in fact predating the ADAS or even pure. And in fact, it was driven by the second PhD student whose work I had talked about last week, Anirban Badri, who had also participated in structural motifs, the beginning of Gendis and so on. However, we decided to zoom in and look at this particular superfamily. And the story actually goes beyond his his inclusion in our lab as a PhD student, because as when I was doing my postdoctoral tenure in Professor Tom Blundell's group, I was coordinating the project which allows us to appreciate multiply aligned sequences of domains that belong to distantly related families, namely superfamilies. And tyrosine phosphatase was one of the superfamilies that I had volunteered to work with. So I had understood a bit of its structure and a bit of its function better. So I suggested this to Anirban when he joined the lab, and he took it up for a large-scale analysis. And at that time, when the structural motifs concept had just been devised, we definitely applied these as constraints, and that is the IMPA. And last week, I was saying how it is much better than the SciBlast, but we consulted both methods and implored into the human genome then available. And this was sometime in 2001, and then the paper was published in 2003, perhaps. But what is interesting, you start with some of the trusted tyrosine phosphatase members as the query, and then implore into the human genome, and then we get a number of hits. And we got, in fact, 141 responses. It suggests that there could be 100 odd tyrosine phosphatases in the entire human genome. And subsequent to validations, which were all computational, we finalized the data set with 96 tyrosine phosphatases. And these belong to 26 domain architectures, as explained by Advait just now. And it clearly suggests that there is a huge convergence, nearly 25% convergence, by means similar sort of domain architectures indeed are being adopted by the paralogs, that is those genes that contain at least one tyrosine phosphatase in the human genome. And how do they look like and how do they vary? So we can apply ADAS, but before that, let me tell you the kind of happiness that one gets by simply going after a pair of genomes, almost like how you do a manual work on the table. So we picked up, for instance, 2 genes across the mouse and the human, and these are the best homologs across these 2 genomes. And these are also called as orthologs, and there's the technical term that was proposed by Eugene Koonin. And this is a powerful computational manner by which you can recognize orthologs. And these are none but the those genes that you see as the best homologs across 2 different genomes. So this gene with best, this as the best homolog, and this gene is going to pick up this as the best homolog across that genome. And then these are termed as orthologs. And this is a very powerful and simple concept. But what Anirban and I had quickly realized was this could be domain-centric. And what appears to be an ortholog across 2 genomes, for instance, could be centered at the PTP, but the other coexisting domains could have lower sequence identity, and some of the domains may or may not be present. And these are not unassigned regions, which we could have clarified using pure. These are simply not there. This particular CHT domain is not there. And if that happens, next our kind of a game was to trace the evolution of this domain architecture. And we can ask very nice questions, such as how many domains are present across a handful of genomes which are far apart in the evolutionary scale, such as humans, Drosophila, worm, and so on. And this particular chart shows us what are all the common genomes. For instance, the one, the box in the center, groups together all the domains that are common between C. elegans, Drosophila, and human. And then the box on the right-hand side would bring in all the domains that are common between Drosophila and human, and so on. So you can see that there is definitely genome-centric analysis. If you perform, there are special genome domains that get recruited for a functional purpose, simply because your environment and the requirement for these organisms are pretty different. So having recognized that there are certain domains that are lost or gained, we could just do some kind of a fun exercise by handpicking a set of genes that believe are orthologs at the central domain, in this case the PTP, and then find out how individual coexisting domains either remain or move away. And for this, the good example is the one that is called as a LAR gene in human genome. And the ortholog for the LAR gene is PTP delta in Drosophila and in C. elegans. And you can see that the overall domain architecture might look similar. In fact, ADAS might be kind enough to give a very low score, low dissimilarity score, because after all, the IG domains are there, the immunoglobulin domains, the fibronectin domains are there, the TM, the transmembrane helix is there, and then there are two PTPs both in the human genome, human gene LAR, as well as in the ortholog in the Drosophila and C. elegans. So, however, if you look more closely, the FN3 domains are have gone through a duplication. And if you take individual domain sequences and try to align with each other, it becomes very clear that the second, third, and fourth domain in the human LAR gene is actually gone through a duplication, and it became the fifth, sixth, and the seventh domain in comparison to what we see in the fly and worm. Likewise, one extra immunoglobulin domain has been recruited in the human LAR gene. So this sort of a fun exercise does happen, and definitely in the case of tyrosine phosphatases, the domain architecture has become more advanced and complex in the human genome compared to their primitive counterparts. And I'm saying primitive just because their genomes are they are smaller and they are compact. On the other hand, the sedentary phosphatases are not adopted very much in the human genome. You will see lesser and lesser numbers of serine phosphatases, and their domain architecture trends are just opposite. The smaller organisms, the primitive organisms such as yeast or even Drosophila worm would have more simpler domain. They seem to have more complex because the yeast has 1, 2, 3, 4 domains, and they seem to be evolving independently. And the cyclase domain seem to have become something else in the human genome, and as well as in worm and in Drosophila. However, the PPM, which is the low molecular phosphatase, has gone on to its own independent trend. So you can see how you can begin by taking a coarse-grained approach of domain architectures and then follow through the evolutionary changes, even among the orthologs, to then identify how much variation sets in and what sort of domains get recruited or shed as we go along. Right. So this is kind of a fun exercise that one can do. And back to the ADAS, which Advait had already explained in the context of the human PTP, this is what appears. You know, in the human genome, in the year 2003, when Anirban carried out the analysis, we were left with about 100 phosphatases, and it gave rise to about 23 different domain architecture. And the ADAS phylogeny on the right-hand side shows that. But years later, in 2017, and then this paper was published a few years later, Tina Bhattacharya in my lab, I suggested her to look back at this. Why? Because not only because the human genome goes through had gone through a large amount of curation in the years, because the first draft is different from how it is now. And sure enough, not only the search algorithms have improved, as I mentioned last week, also the database, the human genome database had also changed. And you can see that there are newer domain architectures coming up. And with this sort of a revised pipeline, Tina then implored into non-human mammalian genomes of 30 different kinds. And even though Tirna is not able to join today, and she's somewhere in the UK as we speak, she has passed on her narration, which I would request Shaila to project. And this will be followed by the fourth story, which is the kind of non-human genomes. Within non-human genomes, how does the TIR domain change over an evolutionary timescale? And that will be narrated by Shaiya. So that's the third, the end of that would be the third end of the third story, and then the fourth story narrated by Shaiya. Shaiya, please.
I'll share my screen. Yes. Yeah, this is a narration by Tina about the protein tyrosine phosphatase.
Genomes, including humans, and recorded several sequences that contained. Hi. So we had looked into 30 mammalian genomes, including humans, and recorded several sequences that contained PTP domains or protein tyrosine phosphatase domains. And we came across a protein called FORM-PD2 that had a PTP domain in only non-placental mammals here, as well as amphibians, reptiles, and birds, but it was not seen to retain a PTP domain in eutherian mammals, which included humans and other organisms such as other model organisms such as rats and mice. And this was very interesting because we also came across one more gene product, PTPN20, which did not have a PTP domain or was not recorded in any of these non-placental mammals. So it can be said that the absence of the PTP domain in FORM-PD2 in the eutherian mammals could be supplemented by PTPN20.
Great. That was a very clear presentation about the variation of domain architectures amongst many mammalian genomes. So the fourth story will be narrated by Shaiya on the TIR domains, likewise in mammalian genomes.
Thank you, ma'am. So today, as ma'am just told, I'll be talking about TIR domains. So these, the TIR domains, they are present in most of the toll-like receptor-like protein and other interleukins protein, which are important for innate immunity. So here's a cartoon diagram showing a TLR4 pathway. So we have adapters like TRAM and TRIP, and for this part of study, I will be focusing mostly on these two adapters, which is TRAM and TRIP. I won't be going into the detail of the pathway, but these are like adapter protein, which mediates the downstream signaling by recognizing the ligand and produces pro and anti-inflammatory mediators. So to look into detail about TRAM, so these are involved in the TLR4 pathway, and TRIF, which is another adapter protein, is involved in TLR4 and TLR3 pathway. These pathways are distinct in terms of the ligands which they recognizes. Like for TLR3, it's double-stranded RNA, and for TLR4, it is lipopolysaccharide. So when we look at the domain level of these TIR domain containing protein, we see that TRAM is a protein which just has a TIR domain. But in case of TRIF, apart from the TIR domain, it also has a TRIF N-terminal domain and a RIM domain. So we were interested to see what sort of pattern are conserved if we see across different orders of mammalia. And so for that, we took different orders and then try looking for the TRAM and TRIF sequences and also compare the TIR domain and use these as our query search to look for further across various genome and see what are the oldest ancestor we can get. So for that, we mined across genome-wide, and then we got sequences from as old as chondritic organism, Coelacanthomorpha, Actinopteri, Amphibia, Creptodera, Crocodilia, Lepidoptera, apes, and mammals. Further, this is a sequence similarity cluster. So here we observe that there were three major clusters of the TRIF and TRAM sequences. And further analyzing these, we recognize that these were clustered based on the organism. Also, if we see each of the cluster and these two major cluster, which is on the left-hand side, corresponds to sequence of TRIF. And on analyzing each of these sequence with respect to the domain architecture, we found that the first major cluster over here has a domain architecture consisting of TRIF, TIR, as well as RHIM domain. But the second smaller cluster over here, which includes most of the organisms from Actinopteri, which is shown in blue, and also Chondrichthyes doesn't have a complete domain architecture as that of the first cluster. So it has TRIF and only a TIR domain. Whereas the third cluster, which is on the right side, has the organism from TRAM protein, and which only has a domain architecture with the TIR domain. Further, we went and took these sequences and constructed a phylogeny analysis where we looked for the TRIF, all the representative from TRIF, all the representative from TRAM, and also we found another adapter protein called as TAG, which is an isoform of TRAM. We tried constructing a timeline and proposed how this would have evolved over time. So we found the TICAM to be an origin of these sequences as old as 684 million years ago, and that later diverged into TRAM, TRIF, and tag-like sequences. Further, we didn't stop there. We further looked at the gene neighbors of these TRAM and TRIF, and we found very well-conserved gene neighbors that do suggest that there are similar functions being conserved from ancestor to the current date mammals. With that, thank you, ma'am.
Great. Yeah. Thank you, Shailya. So with this, we conclude our whole story, to the choice of algorithms like Pure and ADAS helps us to appreciate protein similarities at the domain level. It also helps us to appreciate the evolutionary changes. And I hope we have impressed upon you using these two stories that our objective algorithms are major helpers in broad-based understanding of protein evolution. And also following that, we had shared our story on the tyrosine phosphatases and the TIR domains. And in fact, the TIR domain, we got interested nearly 10 years ago. And another student of mine, Mahita, had started off, but more on the structural end. And that we will get to hear next month. So this month is going to be on sequence searches. So with this, I will stop, and I'll request Shafi to project the publications that had the main publications which talk about some of the works that we had discussed today, corresponding to the 4 stories. And Shafi is also going to tell you what's next in the pipeline, what is the topic in which we will discuss next Thursday at 3 p.m. Yes, that's coming up in the next slide.
Yeah. Hello everyone. So today we heard about 4 short stories, Pure, ADAS, Phosphatase, and TIR. Now what we have given here are the most cited publications in these 4 topics. So first one, Pure. So you heard from Chandrasekhar. It's a web server for prediction of domains in unassigned region in proteins. And the second one, ADAS. So you heard from Advait. So it's an algorithm to compare domain architecture between proteins. Now the third one, phosphatases. So what we have given here, a genome-wide survey publication in human tyrosine phosphatases from Anirban Badri. And there are more recent publication in phosphatases. You can go through our publication list. And the last one, TIR, you heard from Shailya Verma. It's a genome-wide survey again regarding toll or interleukin-1 receptor in mammalian genomes. Yeah. So for next week, we are coming with some interesting topics regarding olfactory receptors, ordering binding proteins, and bioluminescence. Yeah. So we look forward for your presence and stay tuned. Yeah.
Thank you, Shafi. And I would like to acknowledge people who have participated and worked on these projects that we could present as stories. And these are people whose names I kept bringing up. But in terms of the actual names, let me put up these 2 slides, which will tell who are the people. And then the Pure, Chandrasekhar is right here with us, and he presented a bit of the story. And then it was co-guided by Dr. Bernard Hoffman, who's currently in University of Nantes in France. And the ADA story, it was developed by Divya and co-worked with by Advait, and Advait had presented this. And I'm also putting the years here for you to appreciate how things have been moving over time. And on the other two stories. We have Anirban, who started off the work on the tyrosine phosphatases. In fact, Anirban has many more papers in Gene and as well as in Journal of Molecular Biology, which we have not put up. We have just given you some sample publications, and we have recent papers by Tina and Mahita's papers, as I said, was on the structural work. But Shailya has a recent paper. I would urge you to go through them. They will give a lot of details on the data and the results that come, but these are kept as light and informal. So therefore, there we are. And I'm just flashing across some of the group photos which we have been sharing, which we have been gathering for memories. And this is a group photo of the volunteers in a huge conference that we had held in 2010, Asian Pacific Bioinformatics Center. Srinivasan is here in the middle. He was a local organizer. And my Professor Manju Bansal, Professor Saraswati, and I'm somewhere here. And a lot of the students are all over. They are in the blue t-shirts this time. So the red and blue were for volunteers. Okay, so with this, perhaps I should just come out of this slide sharing more, and then we are ready to take any questions. And I'm sorry we've taken a little more time than we planned, but each one of these, I assure you that we have kept it so precise, and the stories actually expand, and you're welcome to read the papers to know what's going on. So right now I will request people like Advait, Shailya, and Chandrasekhar to come on video, and we will also wait for us to look at some of the questions that seem to have come up on the chat screen. For instance, we can see one which says it's by an anonymous attendee. It says, "Can you explain how ADAS tree is constructed?" In between, I was explaining that these dendrograms are constructed exactly the same way people use to create sequence-based dendrograms. That is where they normally supply sequence dissimilarities scores as elements in the matrix. But perhaps Advait has something to say about it. Advait?
Yeah. So in the ADAS, what we get is a domain architecture distance. So that distance is constructed as a matrix, and this matrix is used in the neighbor joining method to get the cladogram. Either it can be neighbor joining or UPGMA, but we prefer neighbor joining, and there are programs such as PhyLib which can very well take this as an input to construct the tree.
All right, great. And there's some question about what's the function of tag protein. If anyone already knew, they could just answer it. But otherwise, I would urge this person to go through the PFAM database or the UniProt database, because there is a very comprehensive record about various functions. And in fact, function of proteins is a very, it's multi-level. So whether we are talking about the biochemistry or the biological process, we have to ask ourselves. If anyone wants to expand on this question, if not, we could go to the next. The key differences between pure and HMM scan. Maybe Chandrasekhar can take this.
Yeah, yes, ma'am. In fact, we use a HMM scan within a pure algorithm. So the HMM scan, what does is given the protein sequence, and there are already HMM models for each domain in the P family. It looks for those profiles and assign if there is a good hit. And that is how PFAM assign domains in their database. But with that direct approach, as I said, only around 50% of residues are assigned to some functional domain. So what we did is we have taken this HMM scan through indirect approach method. So we first look for homologous sequences. For those homologous sequences using HMM scan, we assign the domains. Then we add back onto the main protein sequence with some biological sense. For example, I've shown if there is a transmembrane protein and downstream adenylyl cyclase, there must be a receptor domain outside. So that kind of using that kind of sense and multiple sequence alignments, we can assign domains indirectly. So therefore, we will be able to increase the annotation coverage. I hope that made sense.
Great. Yes. Yeah. In fact, yeah, Chandrasekhar has addressed it very well. And HMM is just one module in the entire Pure pipeline, and Pure is supported by many other elements, which has been carefully included. Thank you, Chandrasekhar, for that. I think Shailya wants to say something about the tag protein, which she has described very nicely, that connects the TRIF and TRAN.
So, yeah, along with that question, the next question follows up how we found tags with TRAM and TRIF. So, considering both the questions, so tag is an isoform of TRAM, so it has a DIR domain. Along with that, it also has a gold domain. So, the function of the gold domain is to reach the Golgi apparatus and then bind to the code protein. So apart from the TRR domain, the gold domain function has this. And if we talk about the overall tag protein, it acts as an inhibitor of TRAM-mediated TLR4 pathway. So there are two types of pathway in TLR4, but today as we are discussing just the TRAM-mediated, and we also found this in our genome-wide search because it has a TIR domain, and it has a lot of similarity in terms of sequence with the TRAM TIR domain.
Great. And there is a question on ADAS algorithm, whether it is freely accessible or available on time. Advait may want to take it.
Yeah, so ADAS is available. It is a simple program written in Perl, and anyone can access it. Please go to the publication, and you can get the script as well, and then you can run it, embed it in your ways as well.
I think we have more or less covered the questions as part. So with this, we would like to thank the volunteers who have been helping with this seminar series, and also the communications office of NCPS TFR for really supporting this initiative. And we look forward to similar discussions, and please stay tuned for further stories that will come in the following weeks. Yeah, like Chappy mentioned. So we meet next Thursday at 3 p.m. Until then, please take care. Bye. Especially Chandrasekhar, all the way from India.
Thanks, ma'am. Yeah, have a nice day. Bye.